To begin this protocol, select a vector for the expression of your favorite bacterial effector in yeast and a yeast strain. The vector is then transformed into yeast cells. Next, expression of the effector is induced. Induced cells are lysed for subsequent Western blot analysis to verify expression of the effector. After confirming expression of the effector, cells are grown, and tenfold serial dilutions are spotted onto repressing and inducing media. Finally, after 2 to 3 days, yeast growth is analyzed.
Hi, I'm Dr. Salomon from the laboratory of Dr. Guido Sessa in the Department of Plant Sciences at Tel Aviv University. Today we will show you a procedure for the identification of growth inhibition phenotypes induced by expression of bacterial type 3 effectors in yeast. We use this procedure in our laboratory to study the function of type 3 effectors from gram-negative pathogenic bacteria and as a tool that assists us in the identification of the effector's eukaryotic targets. So let's get started.
Several important factors should be considered and optimized when designing a yeast system appropriate for expression of the type 3 effectors of interest. The first is the promoter driving expression of the effectors. Since bacterial type 3 effectors may be toxic to yeast cells, an inducible promoter should be used to control their expression. We will use the Gal1 promoter in this experiment. The second factor is the copy number of the effector gene. High expression levels are achieved when the effector gene is carried by a 2-micron plasmid. Intermediate expression is obtained by using a centromere-containing plasmid, and low expression is achieved when the effector gene is integrated into the yeast genome by homologous recombination. A third factor to be considered is the epitope tag used to verify protein expression in cases where antibodies against the effector of interest are not available. Commonly used tags are MYC, hemagglutinin, HA, or FLAG, to which reliable commercial antibodies are available.